Welcome everyone to the theoretical and computational biophysics seminar. After a while, actually, we haven't had this seminar for a few weeks. So this is my great pleasure to introduce Professor Varma from the University of Southern Florida, who is our speaker today. So we were joking that Samir used to be sitting on the other side all the time when he was a biophysics student here, and now he is on this side of the podium. Okay, yes.
It's equally terrifying.
So by way of introduction, Samir studied physics. He got his bachelor's and master's degree. Sorry. Okay. Master degrees from India, and then he came to university to the PhD here in biophysics. He worked with Eric Jacobson, I don't know if you remember. And then he did a postdoc at San Diego National Lab with Susan Lampe, where he studied ion selectivity. And then Samir spent some time in IIT as a research assistant professor before doing a research fellowship at UCLA. And then he joined the Department of Molecular Biosciences at the University of Southern Florida, where he went through the ranks, and he is now a full professor there. So Samir is very active, and actually I was really impressed by the breadth of the systems that he studied, going from ion selectivity and ion channels all the way to viruses, force field development, and machine learning, and so on and so forth. So I was really impressed. And he had a lot of rewards and prizes that I don't want to go through all of them. Multiple awards from USF, ACS, from UCLA, AAAS, and San Diego National Lab. So he was very kind enough when we met in Biophysical Society meeting to accept my invitation and come here for the seminar, because he told me that everything that we are doing with forces that we are using might be 29 kilocalories per mole off. I remember that very clearly.
Yeah, it might be.
Yes, I'm a please.
Thank you for the kind introduction and for an opportunity to come back to Urbana as a professor and not as a graduate student. This is the second time I took a flight into Champaign. The first time I was telling people that I took a flight into Champaign when I got a cheap ticket of $150 from Dallas. This is the second time. Otherwise, I used to fly out of nearby airports. So I got into the air, I was like, man, this is so cool. I had forgotten this was the University of Illinois airport, not Champaign Airport. So let me give you a sense of the things that we are doing in our lab. So you all know
if you ask that
exit.
No, the top of the slide. Yes, check there. Thank you.
And this one.
This one you have to escape, and it's okay. That one. Oh, good, perfect. Go ahead.
Right, and so we all know what these molecular simulations are, and what I don't have to introduce anyone to MD here. And so the thing that we need to keep in mind is this cost of calculation versus accuracy and resolution. Okay, the more resolution you want, the more cost of the calculation. Okay, and so you can go all the way up to gold standard quantum mechanics simulations like coupled cluster theory and quantum Monte Carlo. And all the way down to coarse grain, where your resolution goes down, essentially. And so you have these new machine learning models which are coming up. You may have heard of them. And so these machine learning models are trying to get quantum or DFT level accuracy with the same speed at which you run semi-empirical methods or quantum methods. Okay, so these methods are about 2 orders of magnitude slower than all atom simulations, and they will always remain slower. And the reason they are slower is because of featureization. So you have to pack something up in a different transformation space, send it to ML, and then unpack everything. And so that featureization is the one that is the most expensive one. So these machine learning models, even if they are accurate, get super accurate, they'll only be faster than QM, but never faster than these guys. These guys will always remain 2 orders of magnitude faster. Unless this is what we are realizing at this point. You don't know, a new invention might come up and then the whole thing will change. And Imad and I will lose jobs. At that point, until then, we are good. I don't see any signs of that happening. So,
oh, excuse me, can you share your screen to Zoom people? Because I
no.
think, yeah.
You gotta share this. I don't know how to do it.
What? Okay, let me give the talk. Oh yeah, it was
What do I have
deceptive.
to do? Go to
Zoom, yeah, just make that full screen to Zoom.
use.
Yeah, that's why I think.
It's practice, I think.
Yeah, we use MS Teams.
Oh, I see, Microsoft Teams.
Oh wait, okay, I know.
Yeah,
I know.
told you you have to do it.
I
You're
know,
in
I
charge.
know, sorry.
It has to be.
This is because, yeah, I see my name here and I just didn't. Yeah, okay, can you join as a panelist? Yeah.
Yeah, there.
Can I ask you to log in again?
No. Yeah, and then just. Yes, now to go to the. Yes.
Thank you.
Perfect, thank
It's
you.
working now?
Yes.
Okay. And so some of the work that we have been doing with simulations is, need to get rid of that.
Okay.
The thing that I'm going to present today is enhanced reliability of these molecular mechanics simulations, especially in the case of describing ions. We're also developing QM-MM hybrid models, especially to study post-translational modifications of proteins and how they interfere with protein-protein binding. We're also developing coarse-grained models, especially for fibers. Collagen is what we are working with. We're also developing ML models to speed up quantum simulations. I leave that for a different day. We have developed several methods to analyze big data, such as find allostatic signaling pathways using machine learning. And we're applying this based on how many experimental collaborators we can get. Right? When you want to apply simulations, you absolutely need an experimental collaborator, unless there we are working on ion-biomolecule interactions, protein-DNA interactions, intrinsically disordered proteins, and protein fibers. And so today's talk essentially will be on and so what I'll do is I'll give you a flavor of the development we are doing to a polarizable model that we now call AMOEBA-HFC. And then I'll show you an application of AMOEBA-HFC, where we have used it to understand the role of lithium in neurological disorder treatments. And then I'll tell you what we are doing towards improving non-polarizable force fields as well. So what you learn from polarizable models, you now try to apply to non-polarizable force fields. And so in a simple sense, the way a force field is developed, you develop parameters for ions in water, okay? And then you develop biomolecule parameters like those of proteins, lipids, nucleotides, again in water, okay? And then you hope that you can combine, you can use some sort of mixing rules to describe interactions of these ions and biomolecules which have been developed independently. Okay, for a very long time, we now know that these mixing rules don't work, and so people have come up with a term called NB fix to address that. Okay, now what is NB fix? So the way I explain NB fix is NB fix means you're discarding mixing rules. Okay, that's all it is. Nothing more, just a fancy name for discarding mixing rules. So what do I mean by discarding mixing rules? So when you develop parameters for water molecule, and you have water-water interactions, you have one X set of parameters. Okay. Now when you develop ion parameters, you develop them so that they are. Why is this there? Okay. So you develop them so that these ion parameters work well with water. Okay. And then you go forward and you develop protein parameters, again, that work with water. Okay. And they also work with protein-protein interactions. And then you do the same thing for lipids and nucleotides. Okay. So you get these sort of diagonal forms. Okay. What about all the other terms of how proteins should interact with ions? Okay. You assume a sort of functional form of how you're going to mix those parameters. Okay, you could have an average of Lennard-Jones parameters. You can have an average of polarization parameters. You can use different kinds of averages, and that's what I'm depicting by this predefined mixing rule. Okay, this should never have been done. Okay, in the first place, people should have actually developed exact rules to numbers to combine these different parameters you have obtained from different developments. Okay, and so. NB fix is essentially forgetting what these these functional forms are and developing very specific parameters for. That's what NB fix is, nothing more. Okay, NB stands for non-bonded, so we're just doing the non-bonded fixing. Okay, how well do these non-bonded fix methods work? So people have developed non-bonded fixed parameters for ions interacting with proteins. Okay, and this is work from 2015. From the CHARM group, evaluating how good CHARM is in terms of their accuracy in predicting describing local interactions. Okay, so what you do is you take a structure from the protein data bank, okay, that is bound to an ion, extract the local region around it, about 7 to 8 angstroms, okay, and then you calculate the interaction energy between the ion and the protein. Okay, so that's just interaction energy. Okay, and then you do this using quantum mechanics and your preferred force field. Okay, so here you're seeing the comparisons against QM against CHARM 36 and 2. Okay, and so QM is in red, and your CHARM 36 is here, and this is for calcium. Okay, and look at the scale here, and this is in kilocalories per mole. So the error is of the order of 100 kilocalories per mole. Yes.
Yes, but this only includes the environment, right?
Which environment?
Well, like, oh,
yes.
Yeah, I mean, unless it
This
was
is
done,
towards,
unless it
towards,
was done as a QM and M,
yeah.
then
No,
it's a
no.
fair comparison. If it's just some kind of an energy, then
No. So when you're dealing with pairwise interactions, you're just summing up all the interactions to infinity. That's all you're doing. And so the number of interactions that you add up, you're adding up only up to a certain region. You're not adding the rest of the interactions. It's just a summation. You can do the summation up to a certain point. And beyond 10 angstroms, those interactions get weaker and weaker. Okay, so these are the strongest interactions you're encountering. That's my.
I'm not convinced. For example, with a dielectric response,
Yes.
ion doesn't have to be surrounded immediately by water. It fulfills the dielectric response.
Sure.
So
yes, agree.
But there's
agree.
no water there. Right?
Yeah. So this is just looking at snapshots.
Yeah.
I'm given a certain snapshot. I'm supposed to calculate the total energy. So what am I going to do? I'm going to sum up all the interactions one by one. Okay. And so I'm looking at only a component of those interactions if I sum up to only 10 angstroms.
But maybe that's a bad test.
This is CHARM paper. I'm not doing anything else. Okay, this is CHARM data. Okay, and this is the paper that is published. So all you can conclude from this is that the local interactions are off. There's no other conclusion. I'm not talking about condensed phase this time. I'm only saying, here is my snapshot, here is the interaction energy, it's off by 100 kilocalories for the calcium ion. Okay? The root polarizable model does a lot better, but the average error is 29 kilocalories per calcium. Okay? For sodium and potassium, it's around over 10 kilocalories per. Okay? And then you also have a maximum error that exceeds 55 here, and maximum error for sodium and potassium is 25. Okay, so this is essentially saying what the state of the art is for root polarizable. Okay, and you get the same answer if you use the amoeba polarizable model. You get nothing different. For magnesium, the mean absolute error is 17 kilocalories per mole. You add NB fixed terms, you bring it down to 10 kilocalories per mole. The average error, the maximum error still stays over 25. Okay. Now, the interesting thing is your QM data is here, okay, and this is your amoeba data. Amoeba seems to be underbinding consistently. So there's a systematic underbinding error. Okay, and so this is something that we saw is a sign of probably some physics is missing or misrepresented. And at that point, we can say, let's see if we can fix and find that missing physics. Okay? The other reason we got, and so when we started to think about this problem, there were several sort of potential areas of concern. Okay, the first one was, are we looking at the correct reference target data? Okay, you can have some reference target data from experiments, but it is rare. People typically use quantum mechanical data as their reference, but even in quantum mechanics, there's a huge variety of different methods, and people have used the best method they had at that time. Okay, so when people were developing them in 2003, 2005 timeframe, they were using monovalent second-order perturbation theory, and that was one of the most accurate methods. Okay, and now the most accurate method that is accessible to us is coupled cluster theory with single, double, perturbative, triple excitations at the complete basis set. And we also have quantum Monte Carlo. Okay, so now we can have a higher level of resolution and also accuracy in terms of the reference data. Okay, so it is possible that they were not tuned against the right set of data back then. Okay, is NB fix done appropriately? So are you doing NB fix right? So Alec has his own method to do a non-bonded fix. Okay, is to control osmotic, is to describe osmotic pressure. Okay, so people have, so it's not a set way to do NB fix corrections. Okay, selection of ion parameters. If the ion parameters were selected properly. Okay, were the ligand parameters selected properly? Like I said, all these ligand parameters were parameterized in water. They have never seen an ion. Now you throw an ion in there, what happens? Okay, are you in the correct parameter space? And what necessary physics might be missing, especially in the case of amoeba. Okay, and so at this point, we decided, let's work with the amoeba polarizable. Okay, and so the amoeba polarizable model function is quite complex. Okay, now the way to look at this function is you have these bonded, angle, torsion, stretch bend, high torsion, and torsion. So these are the bonded interactions. Okay, these are taken straight from MM3. Okay, there is no change. So these are MM3 parameters all the way up to 2. Okay, and then you have this multiple expansion. So you have a point dipole, a point monopole, and a point quadrupole, describing each atom center. Okay. Then you have polarization, which is an induced point dipole at the center of each atom. Okay, so you take induced dipole, and that's your polarization. Compared to John Drew uses a Drew particle with attached to a spring and moves back and forth in response to a field. Here it's just a point dipole that is massless particle that is moving there is a field. And then you have these van der Waals interactions. Okay? These van der Waals interactions are described using a buffered 714 function. Okay? Now there is some confusion in the field. If you use 714, it is not 612, it is not a Buckingham exponential 6. But for a given potential, if you want to get this potential in black, you can get it exactly using any of these 3 function forms. You just have to choose the parameters. Okay, so that is that. And so we started working on this amoeba model back in 2017. So we released the first amoeba new model called AMOEBA-HFC back in 2022. Okay, so addressing all those problems I talked about, I'm just going to give you some snippets on what we have done to improve this model and why do we not call it AMOEBA-HFC. Okay. So the first thing was, do we have the correct reference data? Okay, so we determined all of the experimental data that was provided in experiments. This is all mostly Kevarli's data from the 70s and 80s and Descendier's data from the 90s, where you know from experiments the binding energy in the gas phase for sodium binding to water, sodium binding to methanol, and so on and so forth. Okay? And then we determine a density functional theory also that reproduces this data in the gas phase, which is the experimental data, okay? And also reproduces coupled cluster theory data and quantum Monte Carlo data, okay? And we estimated how far is density function theory from these high-level QM methods and from experiments, okay? Now, why do we want density function theory? Is because that is something that we can use to describe 300, 400, 500 particles, okay, or atoms. If you have quantum Monte Carlo, the biggest system we have had has had, I think, 40 to 50 atoms, and that was also pushing the limits of our hardware. Okay, coupled cluster theory, I think the biggest system we have is 65 atoms, and so the idea is to find the density function theory that you can use to go to 500, 600, 700 atoms, but know what the error is with respect to these high-level methods and against experiment. So the PV0 density functional, we know, has an error of about 1 kilocalorie per mole against experiment. Okay. For monovalent cations, it is off by 1 to 2 kilocalories per mole. For divalents, it is off by 1 to 4 kilocalories per mole. Okay. So the best answer that we can give you using your DFT methods as a target will have an accuracy of 4 kcals per mole on the conservative side, okay? And 2 kcals per mole with respect to for monovalent cation. So that is our target, okay? And even if you use DFT, that's the same error you're going to get. You cannot use coupled cluster theory for doing any protein stuff. Okay? So, so let's revisit how these ion parameters are developed. So when you develop ion parameters using non-polarizable force fields and also using polarizable force fields, you try to capture certain aqueous phase properties. Those are becoming your targets, okay? You try to capture hydration free energies, coordination numbers, water resistance time, okay, and osmotic pressure. So those are the properties you try to target, okay? But what about local interactions? Okay, how good are those local interactions? You got the macroscopic properties right. Do you also get the local and distant interactions right? Do you have a good balance between local and distant interactions? Okay, that balance becomes important when you're doing things like an ion leaving water and binding to something. Okay, at that point, you have to strip these waters off and replace them with protein. So your local interactions matter at that point. Okay, and so if you want to get this right, you need to get the local interactions right. Okay, so we said let's flip the whole philosophy of developing ion parameters. Instead of targeting macroscopic properties, let's target local interactions and develop our van der Waals that give correct local interactions, and then see whether we get the correct aqueous phase properties. Okay, and so this is our CCSDT coupled cluster theory numbers. So this is what we target against. Okay, and these are showing what the AMOEBA was. It is off by 6 kilocalories per mole under binding. Okay, 263 versus 255. These are amber 22 off. These are the latest magnesium parameters, 18.2 from World. So they're all under binding with respect to the QM reference data. Okay, what if we fix that and find parameters that do that? So yes, we can find parameters so that the error comes down to 0.6 kcals per mole. Okay. Now, once you find it, you try to predict using those parameters the aqueous phase properties. Okay, and so hydration energy, we get it right. 206, right between the experimental estimates. We are not tuned against them, we're just predicting that. Okay, coordination number, which was easy in the case of magnesium. The coordination first peak, that is where the ligand should lie next to the ion, that also is within the experimental range. And this has always been the hardest property to get is water residence time. That is, how much time does the water spend in the first coordination shell? Now, the way you get that number is by calculating the potential of mean force for extracting that water. Okay, and this rate depends on the peak height. If your peak height is off by a kilocalorie per mole, you'll end up with the wrong answer. Okay, and so we have now not close, pretty close to it. We're in the same ballpark of 1 microsecond. Okay, so this is, I'll show you in the end. This is the only model of magnesium that gets all these properties right. We haven't looked at osmotic pressure.
So you're gonna tell us how you did it, right?
Oh, it's just parameter search for sigma and epsilon.
Oh, okay.
We find a sigma and epsilon that gives you this right. And once you get these local interactions right, everything else falls into place. So this is built inside out instead of building it outside in.
Okay.
It's just a new set of Lennard-Jones that works.
Just interaction with water?
Just water. So all we have are for training are 1-fold, 2-fold, 3-fold, 5-fold, and 6-fold clusters. You just have clusters, okay? And this is just interactions at the local level. All of them are directly coordinated with the ion, and we have multiple geometries. So we try to get both the structure, right, of the optimized geometries as well as the energies. So essentially, we have 12 data points, and we find the sigma and epsilon of magnesium to get those 12 data points as good as you can. So what happens is you end up with 4 or 5 parameters that look good, and then for all of them, you calculate the hydration-free energies and all these properties, and you pick the one that is the closest to experiment. It's an inside-out approach instead of one. So that seems to work, okay? At least for magnesium. Now what you do is you say, okay, now I have my good ion parameters. I'm going to now replace these waters with a different ligand in this case, in methanol. Okay? And I'll calculate this energy change, which I call substitution energy. Okay? And then I see how well do I compare against QM. Okay? So my QM is here, which is in the black line. So as you start replacing waters with methanol, you make the cluster more stable. And that really comes from the polarizability of the methyl groups. Okay? It becomes more and more stable. The old parameters are in red here, and the new van der Waals parameters are red here. So you have the best ion parameter, but you still have this error in substitution energy. Okay, so just because you have a good ion parameter in water, it doesn't mean you get the correct substitution energy. Okay, it's really got to do with finding the correct set of NB fix numbers. Okay, but is NB fix enough? We don't know that because people have tried to use NB fix and they don't get the correct interaction energies. Yes.
So when you're tuning your sigma and epsilon parameters, so I don't know how the amoeba force field is, but like, is it both particles from both atoms? They have their separate.
So water already has its sigma and epsilon.
Yeah. And are you tuning both of them, or?
No, you can't touch the water. The water is already set. You're only training yours. Okay, within the constraints of a mixing group. Okay, so that's another lesson we do. Okay? And then we looked at how does the polarizable model of amoeba respond to electric fields. Okay, so we can place a point charge next to a group. This is just methyl acetate. Okay, you place it next to a methyl acetate, and you determine what is the induced dipole in the molecule in methyl acetate. Okay, so as the distance becomes smaller and smaller, the induced dipole moment increases. Okay, the black is the QM data. The red was the original data, original amoeba model. So you could see it's underpolarized. That is probably, we thought, why it's underbinding. Okay, this is a fundamental change. Okay, so we developed new parameters to get the proper, as good as a response we can get. And we did this not just for one direction here, but for multiple directions. And for all the molecules, functional groups that are known to have cations. The only ones we haven't done are aromatics at this point. We should have done aromatics because recently there was a channel of potassium channel that came out that actually coordinates through aromatics all the way, which was very fascinating to me.
So can I ask a question? So
yeah.
For this curve, you just do it at one distance. You find the right sigma epsilon at one distance.
No, we are not changing. No, this is just a point charge.
Right, but you're searching for, okay. You're searching for the right epsilon and sigma.
No, now we are searching for the parameters of polarization.
Okay, I see.
So we have a polarizable point. Okay, nice. So if you have a point charge, there is no Lennard-Jones interactions anymore. The only thing that you're changing is the polarization response. Okay. Okay, so we should get the correct induced dipole moment in the presence of electric fields. That's all we're trying to do here. So once we get the polarization model, you're like, oh man, the polarization model has to change. And that day I took a really deep breath because now, once you change the polarization model of a small molecule, everything else has to change. the dihedrals, the van der Waals, and everything. And so it took me a while to convince our postdoc to, can you please start on the training of van der Waals parameters? So that's what we did. Okay, so now you have to retrain all the small molecules using for their new van der Waals parameters. Okay, but this is what is typically done. Okay, you take clusters of say NNA or acetate with water. Okay, so we said in our set of clusters that we're going to use to train van der Waals parameters of these atoms here, let's also add ions in there. So let's try to find a phase space that also works well with ions. That was our initial start. Okay, and we did all parameterization simultaneously. Okay, and so which means you have dozens of parameters that have to be optimized simultaneously. And so for that, we used the Mendel-Mead simplex method. And this is just showing the parameter space that we explored for a single set of F sigma and epsilon parameters. So you set a range from 2 to say 5 here or 4.5 for your R. And this is your epsilon, you set a range. And then you run 200 different calculations and you explore the parameter space. And you find places where the RMSE error is small. Okay, so this itself took a year and a half to get all these small molecule parameters. This is a lot of work. So what I'm showing you today is about 7 years of work. It's gone in. And then once you fix the then the Jones parameters, you have to read the dihedrals. Because dihedrals are sort of the dumpsters of force fields. Because there is no such thing as a dihedral. It exists because of electron density interactions. Okay, so whatever is left, you throw it into the dihedral. That's how it works. Okay, and so you retrain the dihedrals if needed. So we check with how good it is. In this case, this was DNP molecule, dimethyl phosphate for the DNA and RNA backbones. Here we had to make huge changes to the dihedral. Okay, and people typically just do it on the 2D energy map or a 1D energy map. We also use structures for this to get good numbers for dihedrals. Okay, and this is again showing you one of these V0 and V1 numbers that will explode, and you end up with something that is in the blue here. Okay. So once this is done, then you see how well your model is performing in the presence of ions. So this is the same substitution energy going from water to NME and water to acetate. The blue is now closer to the black. There was an error that is gone from monovalent cations. And then you also take all these small molecules and you determine their condensed phase properties. So you do liquid NME, liquid methanol, ethanol, and so on and so forth. And what we found was we're not doing any better than the AMOEBA model. Was all consistent with that. Okay, so we didn't break any. Now it responds well at the high field and good at the low field, but nothing is broken in terms of condensed phase. Okay, and then it took us another year and lots of discussions to take these small molecule parameters and put them onto the protein. But now once you have put them onto the protein, we have also changed the backbone parameters of the protein. So once you change the backbone parameters, you have to go and see whether you do well in terms of MD simulations of proteins. Okay, so we tested 8 different proteins. Okay, and so interestingly, so everything seems to be fine. So here I'm also comparing this to CHARM and the A99 SP disk model that works sort of the best for intrinsically disordered proteins. And so you can see for one of these cases, the sigma A1.1 domain, this sort of unfolds in CHARM 36. Okay, but it's actually maintained well in the AMOEBA-HFC model. Okay, this one had huge RMSEs for amoeba, but the AMOEBA-HFC is really good. Okay, and this is the case where we saw the biggest improvement. Okay, this is a GB3 protein. Okay, it's got a unique fall, you have beta sheets and alpha helices mixed in there. This was completely unfolding in the amoeba model. In AMOEBA-HFC, it holds. So we not only, so this was not our target. Our target was just trying to improve the high field response. And it turns out that actually helps also improve the structure of proteins. But that was the coolest result, right, of all of this, that it did well. And then we dug up literature as to how many other force fields have struggled with it. So if you know, the Drude polarizable model came out in 2012 or something, okay? And there's a revised Drude polarizable model of proteins that came out in 2019. And you know the main change that they made there? The main change that they made there was they increased the induced dipole moment coefficients of the backbone atoms to get this right, because the older polarizable group was crashing. And so there they fixed the problem by seeing that it actually falls apart. And here we predicted it by actually fixing the fundamental physics in there. So we were so thrilled that we can get this right even without trying to fix it. So, but there might be cases where it doesn't work, we don't. So in our hands, it has worked really. Okay? And it also improves the dynamics of the amoeba model of the protein. So in terms of dynamics, you look at second order parameters of amino acids. Okay, this is in the case of a protein called ubiquitin. And so the older model was actually more flexible in the loop regions. Now it's not as flexible, and you can reduce the order parameters there. So it has made the model actually more tight and rigid.
So Samir, I noticed actually in your previous slide, GB3 is a stable also in char.
Yes, it is.
Why is it that?
I don't know.
I think you might know a particular reason that explains.
Yeah, we haven't looked into that, but we did look into, has someone else done GB3 simulations? And then we found out, you know, Charm2 had also done this, and the new Charm2 actually works well with GB3. Okay. It was really interesting. And so the bigger question was, GB1 and GB3 have identical forms, but slightly different chemistries. GB1 works fine, GB3 does not.
Yeah.
And it's the same problem that Drew had. So there were certain amino acids that were interacting. So it's the side chains that have problems, not really the backbone. So once you did that, you're like, okay, let's modify this NB-fix protocol also. Okay, so NB-fix is typically done by fixing, you use some QM data to look at how an ion ligand distance changes as energy changes as a function of distance. Okay, you get this profile, and you keep changing these parameters until you can match this. Okay, that's a two-body interaction. But the issue with two-body interaction is it does not have many body effects in it, the polarization effects. So we said, okay, let's just do a many-body fix. Okay, so it accounts for many body effects. And we do this so that it also incorporates interparameter dependencies. So we create some 150 clusters of ions. Okay, and then for them, we calculate their energies and structures, and then we try to reproduce them with those many body terms. Okay, and after doing all of that, you're like, testing time. Okay, and so let's look at this plot. Okay, so this is just showing each of these dots is a structure from the protein data bank for which you are calculating interaction energy. Okay, this was the original amoeba model. You can see the average error was around 17 kilocalories per mole. Maximum error was like 55, so you can find cases where you have large errors. Okay, the amoeba people actually introduced these NB fixed terms in there, and that did bring the error down, but these bad boys were still there. Okay, this is our AMOEBA-HFC without the explicit cross terms that does better. And once we have the necessary cross terms, the error comes down to 6 kcals. And this is what I'm showing here in this color range. Magnesium error comes down to 6. There is still scope for improvements here. And we are now trying to see if we can include explicit charge penetration effects, whether that brings the error down even further. Okay. Preliminary data suggests it brings it down to 4 to charge penetration. So potassium, the error comes down to 2.6, and for sodium, it comes to 2.7. So now this is very close to DFT accuracy. So we have brought it closer, and there is still scope for improvement, at least for local interactions. Okay. So let me take some time, a few minutes, tell you about one of the applications we have done. Okay. So lithium has been used as a treatment for bipolar disorder for over 50 years. It was FDA approved in 1972. We still don't know what the hell it does.
The smallest drug, you know.
Yeah. And so it is a mood stabilizer. It's the first-line therapy for bipolar disorder. It's an antidepressant, the most effective treatment for preventing suicide. It also has preventative properties against neurodegenerative disorders, and is also now a promising anti-cancer agent. Oh, okay. So it does things like in the medical field, people use lithium to pump up hemoglobin count after cancer treatment. Small doses, right? So you give small doses of lithium. So lithium dose should not exceed more than millimolar in the cell. Otherwise, it binds to stuff and then destroys the whole cell. Okay, so it's lethal beyond 2.2 millimolars. That's the average people have estimated. I do not know how they estimated that average. That is lethal beyond 2.2
They have to
millimolar.
measure the concentration in the plasma, actually. If you take in the lithium, you have to do it periodically.
Yeah, you have to measure it. Yeah. And so you have to keep the concentration down in the plasma. And there are no known mechanisms of how lithium partitions from the plasma into the cell. It hijacks sodium channels, which is known to hijack potassium channels, and transporters as well. But it does get in, and it's not selective. So whatever concentration you find in the plasma is the same concentration you find inside the cell. So it just equilibrates. Okay. So what do we know about what it does? Okay. Bottom line, nobody knows. Okay. There are lots of hypotheses. It is known to reduce activities of certain kinases. Of the 75 kinases people have tested, it affects activities or suppresses the activities of, I think, 5 kinases. And by suppressing it, it just lowers the activity by half. That's all it does at 1 millimolar concentrations. Okay. There have been extensive GWAS studies. Millions and millions of dollars were spent on GWAS studies. And what did they find? They found only one thing, that there are in some factions some non-coding RNAs which are different in people who respond to lithium and people who don't respond. Okay. There's also a hypothesis that lithium binds to ATP and disturbs in ATP. How, we don't know that. Okay. And finally, people have found there are certain enzymes that are actually ATP activated transporters that are affected by lithium binding to ATP. Okay. But how does all of this happen? Okay. Central to all of this is the hypothesis regarding the bioactive form of lithium. So when lithium goes into a cell, what happens to it? Okay? And what is known from NMR experiments and other experiments as well is that lithium binds to ATP. Okay? And so what we know is magnesium, wherever you have ATP, there is magnesium bound to it all the time. Okay? So you have magnesium bound to ATP, and we know that the binding free energy is around 8.5 kilocalories. So it binds very strongly to ATP. Where does it bind to ATP? There have been 13 NMR studies, and none of them agree with each other. Okay, that's what we know about magnesium. Okay? What we do know about lithium from NMR is that lithium binds to magnesium-loaded ATP, and it has delta G of 3.8. And look at the Kb, which is 1.7 millimolar, which is just higher than the lithium concentration. Okay, and so it's a problem. Okay, so now if you assume the Ng-ATP concentration in the cell to be between 2 to 10 millimolar, you'll see that most of the lithium would be bound to ATP. If you just use this information, nothing more. Okay, the problem is in the cell you also have sodium and potassium, and if lithium binds, why doesn't sodium and potassium bind? Okay. And so you actually have a soup of ions here. Okay? So you have ATP, which is in this range of concentrations. You have lithium here. You have sodium, which is around 15 millimolar. And you have potassium, which is 150 millimolar. Okay? So how do you now tackle this problem? Okay? So the first thing we did was we used MD to find out where magnesium binds. So again, this is just repeating all these force fields that we fixed for ATP. We developed this force field for ATP, but it's the same protocol. It has the best numbers we have for the local interactions. Now I'm going to show you numbers whether we can produce condensed phase properties as well. Okay? So the first question was, where does magnesium bind to ATP? Okay? Now there are 15 possible combinations. Okay? So this alpha phosphate, beta, and gamma phosphate. So if you combine these, you get 15 possible combinations of where magnesium can bind. Okay? So for example, if you have this alpha beta gamma interaction, you can have this alpha beta interaction, you can also just have an alpha interaction. Okay, so there are multiple ways in which it can bind. So we said, okay, let's create geometries in these coordinations and put them in a box of water with very little ions and put the restraints on, keep them for, I think, 15 nanoseconds, and then release the constraints and see where the ion goes from there. Okay. So this is what we did. So actually not 15 plus 3 nanoseconds, you have this restraints and then you remove them slowly. And then where you see where the ion goes after that, okay? And what we found out was of those 15 states, there were only 7 stable states in MP. Okay? Now, just by looking at the distribution of these ions, you can see that this should be the tightest and the strongest binder, and this guy should be the weakest binder. Okay? And now you use something called a double decoupling method to calculate the binding free energies in each of these states. Okay? And you get numbers which are written on top here. And so again, for this one, the binding free energy was 9.06 kilocalories per mole. This was the weakest, 0.39 kcals per mole. Okay? So then you do a Boltzmann average of all of these states, and this is the predicted binding free energy of magnesium to ATP. And these are 36 experiments clubbed in different experiment types. And that's the range of these experiments that were done. So we fall within the range. We have not tuned for this, but this is a prediction. Okay, it's just making sure that the local interactions are good, gives you those condensed phase properties. Okay, now we do the same thing for lithium. And for lithium, we find there is only one state. And it is this one, a combination of alpha, gamma, alpha, gamma, gamma, alpha, alpha. So it's just this alpha and gamma position. It's actually the same position where magnesium binds, right there between the alpha and the gamma. So it's like this, magnesium binds in here. If there is no magnesium and you put in lithium, lithium won't bind. Okay. Now, and this is the free energy we get from MD. This was the experiment. It's fairly close. Okay. And now we see whether we can reproduce the NMR data that showed that the binding free energy in the presence of magnesium will be actually stronger. Okay, it's some cooperative effect, and it does. And so in the presence of magnesium, it actually binds right next to it. So if you have your alpha, beta, and gamma ATP phosphates, magnesium binds in the middle, and lithium could bind either on this side or on that side. They are chemically equivalent sites. Okay, and when it binds there, it's ND, so it's 3.7, and experiment was 3.8. So this was a surprise. So really close. And at that point, you tell your graduate student, do it again. Do it again. You must have done something wrong. You can't get something this close. I was okay with 1 kcal off, but you give me a number like that, you got to do it again. Okay. So we did that again. And then we determined where sodium and potassium binds. So sodium and potassium bind exactly the same place where lithium binds. Okay. Sodium binds a little weaker, and potassium binds even a little more weaker. So these are our predictions. There are no experimental use for it. Okay. And so you have lithium, potassium, and sodium binding at the same site. This binds the strongest, sodium the next, and potassium the weakest. And the concentrations of these guys are in the opposite direction. Yeah. This is the highest concentration. This is 15 millimolar. This is 1 millimolar. What do you think would happen? So we did about 15 microseconds of MD under various different combinations of these concentrations. And this is what we learned from this.
Okay. Using your own force field.
Yeah, it's an AMOEBA-HFC, right?
Okay.
Yeah. These are, I think all simulations were 600 nanoseconds long. Nice. And we did many of those under many combinations to find out what's really happening. Okay. And so what we saw was, this is just one of the 600 nanosecond simulations. And here you have about 150 millimolar potassium, 15 millimolar sodium, and 5 millimolar lithium. It's in a higher level. Yeah. But the box size we had, 5 millimolar means 1 ion. Yeah. of lithium. That's what the 5 millimolar means. If you had to go 1 millimolar, we had to go 5 times the size and not forget. Right? And so what we see is that the green ones here, the different shades of green are the different potassium ions that are coming. Okay. This 0 is where the magnesium is bound and shown today. Okay. These are the two adjacent chemically equivalent sites. Okay, which are this side and that side, plus and minus. Okay, so what you see is constantly bombarded by potassium. It binds, stays there for a few nanoseconds and leaves, and a different potassium comes in and bombards it. So it's constantly being bombarded by potassium. Okay, so sodium also comes in back and forth. It stays for some time and leaves. lithium comes in, stays for a longer time, okay? And this stays for a longer time lithium here, okay? But magnesium does not get displaced in any of those 15 microsecond simulations, as you would expect, because it has binding affinity of around 9 kilocalories per mole, and all these other ions have 2 or 3, okay? And so what we see is that there's a huge uncertainty. You only see actually 2 lithium binding events. And those 2 lithium binding events are shown there. So that's why the number of binding events is 2 plus minus 2. Okay? We see about 57 sodium binding events, okay? And an average residence time of around 5 nanoseconds. Potassium, we had 665 binding events with an average lifetime of 0.7. Okay? Now, this is the kind of statistics that makes you worry. I can't do any more MD. I have to do something else. Okay? So what we did was we constructed a kinetic model. Okay? So there are 3 binding sites in here. Okay? So we constructed a 3-site kinetic model. Okay? We know what these Ks are. They are related to the Kds. Okay? So the K on rates, we said for lithium, let's set the lithium K on rate to be 1. And the sodium and potassium K on rates would now be a ratio of the ratios of their diffusion coefficients. Okay, so they are related to the diffusion coefficients of their on rates. Okay, and the off rates then are automatically the on rates divided by the KD that we get. So this is settled. Okay, now the other thing we noted from these simulations was that the average ion occupancy was 2.39. Which means one side is low, if magnesium is loaded on one, a monovalent ion is loaded on the other, then the probability of finding the third site is not going to be 1, it's actually going to be 0.39. So there is a cooperative, anti-cooperative effect that is preventing the third ion to bind. Okay, so it's not always loaded with 3 ions. And so we introduced this F factor here in the KDs of these guys. For going from 2 ion loaded state to 3 ion loaded state. And we calibrated that F to get the correct occupancy. So if you choose really high F, you can get a low occupancy. If you choose F that is 0, actually 1, you get an occupancy of 2.8. So we check the occupancy based on this. Okay. And then we ran the model. And this is what we found, which was surprising to us, is that in the absence of lithium, this is the probability of finding these different states. Okay, so this line here is essentially ATP bound to magnesium. In site 1, you could have sodium and potassium. In site 2, you could also have sodium and potassium. So about 93% of ATP is always loaded with either 1 magnesium and 1 sodium and potassium as another sodium and potassium. So the bioactive form of ATP is not ATP bound to magnesium, but ATP bound to magnesium and 1.5 sodium and potassium ions. That's our proposition. I don't know how to test this experiment. Okay, now what happens when you introduce lithium? Okay, now what I'm showing you is the percentage of lithium that is going to be bound to MgATP. Okay, as you increase the MgATP concentration, okay, the amount of lithium that gets loaded increases. Okay? If you do this in the absence of sodium and potassium, you have much more loading of lithium. But in the presence of sodium and potassium, the amount of loading actually goes down. Okay? Now, if you are in a cardiac muscle, you will have both states of lithium available. The hydrated form will be around 60%, and the ATP bound form will be around 40%. Okay? But if you're in the neuron cells, that ratio will be small. So around 20% will be bound ATP and 80% will be hydrated. Okay, so that gives us now bioactive form of lithium that exists in both states, and that hopefully should get accepted in DAX. We got reviews back and nobody complained.
Okay.
So I was like, okay, that happens sometimes. So we sent it to Nature Chemistry thinking we found something good. And within 3 days, they sent it back, no, you didn't. Yeah. So, and on top of that, the editor sent me a paper to review a week later. Like, burn! So I reluctantly accepted that paper, but that's okay. But the reviews came back nice last week. So this should be so like, okay, that's good enough. So that is the time when you feel, your PhD student will spend a lot of time developing these methods, and then they do an application, it goes to a good journal. My job is done. You can be out of here with a good paper. Done. So that's sort of the PI's dream. So once you've done really good, towards the end, you have a good paper. My job is done. So I felt good for that. Okay. So the last topic I'm going to spend.
So before you move on, actually, what is the mechanism of lithium based on this thing? So it binds ATP and somehow messes with the interaction of ATP with other things?
No, so now we know what the, now if you want to see where lithium binds to kinase, for example, you're not restricted to the ATP binding site. Because what was previously proposed was that all of the lithium is going to be bound to ATP. So everybody was assuming that the lithium binding site in kinase is going to be next to ATP.
Okay.
Now we can look further than that. We are also exploring that lithium might actually be allosterically binding.
Yeah, but I mean, if you don't have lithium, either potassium or sodium are going to bind to those sites apparently, right?
To ATP, yeah.
To ATP.
Yeah. ATP, so, not necessarily to kinase.
Right.
Okay. And so one of the cool things there, since you brought it up, is that when people study in experiments, they go to the lab and then they look at kinetics of a certain kinase or any enzyme that uses ATP. They do it in a certain buffer salt concentration, which is PBS buffer, which is around 25 to 50 millimolar salt. At that point, when you want to take ATP from solution and put it in your catalytic site, you probably only have to get rid of one extra monovalent cation or half monovalent cation. But if you do it at cellular concentrations of 150 millimolar, you actually have to get rid of 1.5 ions, which is going to cost you around 3 kilocalories per mole. So now what you're going to do is, as you increase the salt concentration, what you should see is that your enzyme catalytic activity should go down, because it costs you more to recruit the ATP and strip it off its monovalent cation. That's what we are seeing. And so I've talked to a bunch of experimentalists about this, and their whole reaction was, oh, that's why it was happening. Nobody has come back and told me you're wrong. So these are all private conversations. So we were recruiting a structural biologist for our department this year. So we had 6 candidates who visited campus, and to all of them I showed this, and all of them were like, yeah, that's why this happened to my experiment. So either they agreed with me because they were interviewing, I don't know. Right? So probably. But if you tell Claudio, he doesn't care. He just give it to you straight away. So I don't know. I'll talk to you more about this later.
Okay, I'll let you go.
Yeah. Okay.
So much time actually.
Yeah. This is the last part. So what are we doing now in terms of what can we learn from this to improve non-polarizable force? Okay. So what I'm showing you might look complicated. There are lots of dots and numbers. So what we are doing is comparing the errors in modeling magnesium water interactions. Okay, we have non-polarizable force fields as circles. Polarizable force fields are stars. Okay, and there is also this 1264 potential from Kenny Merz. That is a square. Okay, and these are errors. So the closer to 0 you are, the better you are. Okay, there's only one force field that you see that is close to 0 here and in this plot. And this is ours. Everything else is followed. Okay. Now, how different they are. If you look at non-polarizable force fields, you see this nice little trend. And this is ion-water distance, and this is hydration-free energy. If you want to get the hydration-free energy right, you have to have an error in your ion-water distance of around 2.2 angstroms. So you have to get them closer. And the reason you need to get them closer is because you don't have polarization energy coming from the rest of the system. But if you compromise on the hydration-free energy, you can get the correct ion-water distance. Okay, polarizable models don't have problem. The 1264 also does well. You're getting both of these together. Okay, on here you have these local interactions. We tuned these local interactions, so that is why we are here. Okay, they're close to 0. And this is residence time in order of magnitude. Okay, so we are within the order of magnitude. And if you pick any of these polarizable models, they are off by, this one is off by a factor of 3. 1,000-fold difference. Okay? And this is off by a factor of 10. Another polarizable model. Okay? And if you try to get this right, your local interactions are messed up. Okay, this is the only one that gets it. This is the growth set model. This is messed up, and the local interactions are messed up. So you get the correct angle distance. So you can get all 4 of these right. Okay. Now, this is something we were talking in the morning with Alan. So if I put osmotic pressure and make another one way to this line, and this is something we haven't done yet, but we would like to. But for biological purposes, for magnesium, we don't want to go beyond a few millimolars of concentration. So this is fine. You have to deal with osmotic pressure when you do 1 molar of magnesium. So this is okay for bio applications. Okie dokie. Let's go for it. Okay. Now, if you if you run a molecular dynamic simulation of a magnesium ion in water, okay, say a 10 nanosecond simulation, and you extract 50 snapshots from it, and you only extract the inner shell of waters, just 6 waters out. You close the 6, and you calculate the interaction. Okay, this is AMOEBA-HFC. This is charm, and this is YAM. So what you do is, what you find is that you are over binding in the first shell because it gives you the correct hydration, and that's what I said earlier. But you're over binding with 60 kilocalories, right? So it's in the inner shell. Okay. Now this is magnesium protein. I had shown you this before. We get this pretty well within 6 kcal per mole. This is for protein PDB. This is one of them is charm and one of them is amber. Okay. You have large possible errors. You could have a large 80 kilocalorie plus, or you could have an 80 kilocalorie minus. You don't know where you are. Okay, you could be off this way or this way. Now, if you're off this way, you could cancel out these errors, and you could calculate the ion binding ability correctly. So you might be in a state where you can do that. Okay, so this is what we actually did to a non-polarizable force field. We simulated a non-polarizable force field, which was actually Eric's Jacobson's lipid polarizable force field, and we adjusted these parameters using what we call the substitution rule. Okay, so our recommended solution is don't try to get the interaction energies right for the protein. Try to get the relative energies with respect to water right, so you get rid of the error with respect to water. Okay, so you always train with the substitution. This is our recommendation. You know, that's what we made in 2020 to offset that error with respect to water. Okay, and then we applied to a lipid system. So if you have done lipid simulations, I know some of you have. If you take a POPC bilayer and you simulate the POPC bilayer without salt, and then you add 100 millimolars or 150 millimolars of potassium chloride, you will see that the membrane actually, the area per lipid increases, right? No, the bilayer actually thickens. Area per lipid actually decreases, and it thickens. This is something you see from all ND simulations that have been run so far. There's a lipids database, simulation database in Europe, you can see that. And no one has gotten that right, okay? It's because sodium interactions with, or potassium interactions with lipid were not fixed. So we use the substitution energy thing, and we actually get the right answer. You get closer to it, not the exact answer, okay? So this is black, again, is our QL data. This is what was before we did the fixes. And this is where we fix the substitution energy in red to get the correct substitution energy. And then we did lipid membrane simulations and determine the form factor. So if you look at this plot, there are 4 plots in here. You have the experiments without salt, which is this one, the gray line, okay? And so if you do the experiment with salt, if nothing changes, the form factor remains the same, okay? And when the form factor remains the same, this first intercept here tells you about the width of the bilayer, okay? So what it's telling you is that this old parameters, this blue one, shifted this. So it's increasing the packing. Okay. Now, when we use our optimized parameters, we shift it back towards the black, but it's not all the way. It's gotten better, but it's not all the way. So there is an improvement, but not all the way. And so now we are trying to see if we can develop polarizable models for the lipids and see if we can get this. We are trying this approach again as of now to improve these cross terms for charm and other forces. So this is where we are at this time. And so my take-home message essentially is that improving ion interaction is actually more about getting the ligand right, and not necessarily just getting the ion right. Classical polarizable force fields can achieve QM level accuracy. And so what I would do is I would make this go here. This is what we are trying to do. If you can do this, even if the ML model comes, we'll still be 2 orders magnitude faster. That's one of the take-home messages. And for ATP, its active form is bioactive form is when it's bound to sodium and potassium, and lithium exerts a therapeutic pressure. The AMOEBA-HFC force field actually is available when you download the Tinker package. It's available in Tinker. When you download it, it comes with it. So, and these are the folks who have contributed specifically to this project, a bunch of postdocs and students in about 7 years. The thing that goes without saying is people who develop software. People always forget who develops software, but this is the core of everything. Yeah. Right, so we have used Tinker and Gromax. We also contributed to Tinker software development. And then we have these multiple QM packages that we use, and the optimization algorithms, which is called PAROS. And this is our kind of group. And thanks to NIH and for keeping us on. Thank you all for listening.
Thank you very much, Tammy, for the very interesting and provoking seminar. I hope that the students learned a lot about the power of fundamental calculations. Questions? Any questions?
A comment about this modifier. Now, 1 molar, 1 millimolar.
Yeah.
In our hands, if it's by a factor of 10 off at 1 molar, it's also by a factor of 10 off at 1 molar. Okay, it's linear.
Thank you for that. Thank you for that. I will. That's why. No. That's important. Now I'm not an osmotic pressure kind of guy.
It's always.
It's always the straight. Okay. We'll take a look at it.
Yeah. I had a question about
that is very cool.
Binding to 2 more ions in addition to magnesium.
Yeah.
Right.
Yeah.
So did you see this in any crystal structure, or you think they're stripped off?
They're stripped off. In the crystals,
they bind the protein, they come off.
They come off.
But I've never seen it.
I've never seen it, right? And so that's where the energy penalty comes in, including ATP. That's what we think is happening. And so it has to get rid of those sodium and potassium.
And how expensive is your force field calculation compared to the classic structure type of?
An order of magnitude slower.
An order of magnitude.
Yeah. So
time.
It's not enough. Because there are 2 versions of Tinker now. One is standalone GPU version. You can run it on a single GPU. And that's the one we use for free energy calculations, because when you want to spread it over 21 calculations, each one runs for 60 nanoseconds each. And that's easy, you get the answer in 2 days.
I was actually really impressed by your magnesium data. So how is the, I mean, you mentioned water resistance time is difficult, but do you get a close enough water exchange around magnesium?
Yeah, it's within a micro. So the residence time of a water is 1 microsecond.
Yes.
And that's what we get.
Oh, you get it.
Yeah, everybody else is on. Yeah. So if you run it for 10 microseconds, you can expect to see exchanges.
Any other questions? If not, let's thank Samir one more time. Very good, actually. Very nice. Thank you. You earned your dinner.